2, 1, drop it. Are you ready for this?
Would you be interested in buying some Cram? I'm selling multiple Crams, very different flavors, yes. I'm selling raspberry Cram, berry Cram, very berry Cram, only 20 gold coins for 1 bucket of Cram. What is Cram, you may ask? It's a little mushy substance you put between your gums. It's not dip though, don't think it's a dip. I recommend the very berry Cram as it's like the berry Cram but very. You will receive Cram upon paying me 20 gold coins, yes. For 50 gold coins, I can make a Cram sword. Laid out in the sun for a fortnight, it is stronger than steel. Head to Goop.Store to buy Cram for only 20 gold coins. That's Goop.Store for Cram.
Hello everyone and welcome back to part 2 for lesson 1 module 2. In this part of the lesson we will be looking at specialized cells, what they do, what their features are and how they are made. So to get started on, specialized cells are the foundation of advanced organization in multicellular organisms. So multicellular organisms contain many different types of specialized cells, and all these cells work together to ensure the organism survives. Now, specialized cells have unique actual jobs within the organism. Taking, for example, a red blood cell, its job is to transport oxygen. Striated muscle cells, their job is to aid in the movement of the organism. And nerve cells, their job is to signal changes in the environment. Now, what we say in biology is that specialized cells divide the work of an organism and are structurally designed for their specific job. This allows these specialized cells to carry out their job effectively so that the organism can survive better. So let's unpack that and look at the red blood cells as an example. Red blood cells have the job of transporting oxygen. Now, they've evolved or are designed to actually do this job really well. If you look at the structure of a red blood cell in your textbook, you're going to notice that it's kind of fat on the outside and then in the center it becomes really thin. Now, that is a specific design for the red blood cell. It's evolved to be that way. That's because that thin section in the center is where oxygen can diffuse easily out of the red blood cell into a nearby cell, therefore allowing the red blood cell to easily transport and deposit oxygen around an organism. Likewise, nerve cells that signal changes in the environment are structurally designed for this. A single nerve cell is called a neuron, and if you look closely at this picture, you're going to notice these web-like protrusions or parts coming off the neuron. Now, these are called dendrites, and they allow the nerve cell or the neuron to bind or stick to other nerve cells and communicate with them, or receive communication from receptors. So it allows the nerve cell to receive a lot of input from all different parts of the body and then funnel it into the brain. So it allows it to effectively signal changes from the environment. Now, with specialized cells, we say that there are 2 major features to them. The first is what we call unique proteins. Now, every cell in the body contains the same fundamental proteins that allow it to function. So every cell will have proteins responsible for breaking down glucose and enzymes that will help in that process. However, when a cell becomes specialized, it begins to produce proteins that are only found in that specific cell. I'll give you an example of that. Hemoglobin in red blood cells. So hemoglobin is a large protein found only in red blood cells and allows the red blood cell to bind oxygen. Now, the red blood cell's job is to transport oxygen around the body. So it has a unique protein that allows it to do this job. Other specialized cells also have unique proteins that allow them to do their specialized function. Hemoglobin is just a really nice example because you will be talking a lot about transport around animal and plants. And so hemoglobin for animals is a great example of the unique protein found within this specialized cell. The other major feature of specialized cells is their unique structure. Now, specialized cells aren't all the same shape or size. They actually have a very specialized or specific external structure. And this is because there's a relationship between the structure and function of the cell. Let's look at an example of this one. And if you can understand this example, it's actually really a good one to use in your exams. This is a single nerve cell called a neuron. Okay? You have your cell body in the center. Coming off this nerve cell are the dendrites. We mentioned that in the last slide. Now, these dendrites allow the nerve cell to communicate with other cells around it or receive input from receptors. Now, if the role of a nerve cell, or a neuron in this case, is to signal changes from an environment, let's think about our hand. How many touch receptors must be in your finger, and how many nerve cells do you think there are per touch receptor? It wouldn't be efficient to have one nerve cell per touch receptor. That would be a waste of energy. So the unique structure of the nerve cell means that it can actually have multiple receptors feeding into the one body, and then this can relay the message to multiple nerve cells. So you may have thousands of touch receptors in your finger, but only 10 or 15 nerve cells to take the message from your finger and then relay it to another nerve cell that will then cascade it all the way to your brain to tell you that what you're touching feels a certain way, or if it's a heat receptor, what you're touching is hot or cold, etc. So nerve cells have a very unique structure where they can actually take in a lot of information and then channel it into the central nervous system. Another good example of this will be the epithelial tissue found within your small intestinal tract. Now, if you remember, the function of your small intestinal tract is to absorb nutrients from the food that we're digesting. Now, you're going to notice on the top of the epithelial cells, which is the kind of layer that comes in contact with the food that you're digesting, there are all these really kind of hair-like structures called microvilli. The reason why the epithelial cells in the small intestines have these microvilli is to increase the surface area with the nutrients going through your small intestines. That will increase how much or how many nutrients diffuse from your small intestines into your cell, then into your bloodstream. So we say that unique structures between specialized cells allow them to do their job better. This may be an increase in surface area to volume ratio, like seen in the epithelial small intestinal cells, or it will allow for large amounts of information to be communicated across long distances, as is the case of our nerve cells or neuron cells. So these are the kind of major features of specialized cells. They have unique proteins and unique structures that allow them to do their job super effectively and allows the organism to survive. Now, these specialized cells need to be created. When an organism is first forming as an embryo, there are no real specialized cells. They need to be made from something. So in biology, we've found that specialized cells come from a specific type of cell called a stem cell. Now, stem cells contain all the basic metabolical processes for survival, and they also contain the entire human genome, as does all our cells. However, these cells are considered blank slates. They are not specialized in any way, shape, or form. So the definition of a stem cell is the following. Stem cells are undifferentiated cells that can be turned into specialized cells, causing it to produce specialized proteins and developing a specialized structure. So your stem cell is a blank slate. It can then be turned into a specialized cell where it begins to develop its unique proteins and unique structure. Now, I've used the word here undifferentiated because the process of a stem cell being turned into a specialized cell is known as differentiation. When we activate them into a specialized cell, we are differentiating them. We're turning them into a specialized cell. So the differentiation, or the definition for differentiation, is the process of turning a stem cell into a specialized cell by exposing it to hormones and chemicals that initiate specialized protein production. Now, it's important to note that once a stem cell is differentiated into a specialized cell, it cannot change again. So you can't take a stem cell, differentiate it into a nerve cell, and then bring it back into a stem cell. That doesn't work. Nor can you take a stem cell, differentiate it into a nerve cell, and then try and turn it into a liver cell. That doesn't work. Once it's specialized, it remains as that specialized cell and will go through mitosis and divide into more of that same cell. Now, I've got a diagram that may help kind of break this all down for you. There's a lot of specific biological language that's being used here. But just in short for you, stem cells contain all human genomic material, all genomic material for that organism. They have the metabolic processes for survival, but they are blank slates. They have no specialized features. Stem cells are considered to be undifferentiated. We can expose stem cells to chemicals and hormones to start a process known as differentiation, which is the process of turning a stem cell from its blank slate into a specialized cell. Okay, and once you've done that, it can't go back. So let's look at this diagram, and hopefully this will help kind of break it down for you. We have our stem cell. It contains all the genetic material for the organism. It also has the key metabolic processes for survival. Now, our stem cell can be exposed to hormones and chemicals. Now, when it is exposed to hormones and chemicals, the genetic material stored within the stem cell switches on, but only certain parts of it switch on. So, for example, route A, the stem cell is exposed to neuron growth hormone. This switches on neuron-specific genes. Now, if you remember, genes lead to proteins being produced. So if you've got the neuron-specific genes switching on, you're going to get neuron-specific proteins being made. As these proteins are made, this stem cell differentiates and changes into a nerve cell, a neuron. Now, this neuron has the unique proteins that were produced by the neuron-specific genes and has developed the unique structure through the process of differentiation. Now, if this neuron was to divide through mitosis, it would give rise to more neurons. It doesn't create more stem cells because once a stem cell is differentiated into a specialized cell, it can no longer go back and it no longer produces more stem cells. So if this neuron was to actually divide through mitosis, it would make more neurons. Another example of this, route B, if we had the same stem cell but exposed it to epithelial growth hormones, so epithelial being surface cells, so in this case maybe a small intestinal cell, if we exposed it to epithelial growth hormones, the part of the genomic material for the organism would be switched on for the epithelial specific gene. This would give rise to epithelial specific proteins. Now, these epithelial specific proteins would cause this differentiation to occur, and you would actually get an epithelial cell forming from this stem cell. So the stem cell would differentiate into an epithelial cell. Likewise, if it underwent mitosis, it would divide into another epithelial cell and not into a stem cell. Hopefully this image has helped kind of break down how specialized cells are made. They do come from stem cells, and those stem cells turn into specialized cells when exposed to certain chemicals or hormones. And this happens by part of the genetic material stored within the stem cell becoming active. And it's important to note that every cell contains all the DNA for the organism. So it's a matter of these hormones basically saying this part of the DNA needs to switch on now and not these parts here. Whereas in route B, epithelial growth hormone would say the epithelial genes switch on. Route A would say the neuron genes switch on, and that gives rise to those unique proteins that are found in each specific cell. Now, where these stem cells are found in animals does vary. It changes depending on the, uh, age of the organism. So from infants or sorry, eggs all the way up to adulthood, it will change where these stem cells are found and the types of stem cells found. So to start off with, multicellular organisms begin as a single cell. Okay, and this is called a zygote. It's the fusion of an egg and sperm. Zygotes rapidly divide to create embryonic stem cells for differentiation into specialized cells. So here you have your zygote forming, and they rapidly divide into what's called a blastocyte. Now, stored within the blastocyte are these what we call pluripotent stem cells. Now, these pluripotent stem cells can be differentiated into any specialized cell. The word pluripotent means it can be differentiated into anything. So pluri meaning plural, multiple. So it can be differentiated into multiple different specialized cells. So when we have our zygote, it rapidly divides into a blastocyst where you get your inner mass cells, which are pluripotent, and then the mother or the organism carrying the blastocyst begins to feed hormones or expose these cells to various hormones, causing the differentiation of these stem cells into different types of cells. Now, in developed animals, post-embryo, you no longer have these pluripotent stem cells. Instead, we have what's called multipotent or unipotent stem cells. Now, it's generally found within bone marrow and brain tissues. Now, these stem cells here, multipotent and unipotent stem cells, can only differentiate into a limited number of specialized cells. So, multipotent stem cells can differentiate into a few different types of specialized cells, whereas unipotent stem cells can only differentiate generally into one other specialized or one or two other specialized cells. So to kind of give you a better idea of it, these are the types of stem cells and what they can differentiate into. So with this image here, the totipotent cells are the ones that you see during the whole rapid development of the zygote. This leads into pluripotent cells. So these are the ones found in the blastocyst, and they can form over 200 different types of specialized cells. Now, this is how that embryo begins to form a heart, brain, etc. So the mother that is carrying the organism or the seedling basically starts to expose these pluripotent stem cells to various hormones to begin to create cardiac cells, uh, nervous tissue, bone, etc. Now, in adults, so post embryonic phase, we no longer have these pluripotent cells. We have what we call multipotent cells. So these are the cells that can differentiate into a number of different tissue types, but it's much less than the pluripotent. And this is generally found in our bone marrow and nervous tissue. And so this image here is just from your textbook, kind of trying to show you that um, with pluripotent, you get different types of cells being formed, whereas multipotent is more specific. So the multipotent stem cells found in the bone marrow can become heart muscle or red blood cells, but nothing else, etc. Now, in plants, the same process does occur. However, where you find the stem cells is different. So in mature plants, the stem cells are found in the meristematic tissue, and this is the root tips. So the tip of the root of the plant contains undifferentiated stem cells, and they can then differentiate into phloem, xylem, root hairs, epidermis. So the actual plant itself stores its stem cells right at the tip of the root. So what you've looked at today is the organization of multicellular organisms, as well as the foundation to how these multicellular organisms are organized, and the foundation of that are these specialized cells. To give you a quick recap of what we have done today, you've looked at the difference between the organization in unicellular organisms, multicellular organisms, and colonial organisms, and you've got a bit of an appreciation of why multicellular organisms require specialized cells and why individual cells from multicellular organisms cannot survive on their own. We then looked at the organization, so cells to tissue to organ systems, and discussed how these function together to support the life of a more complex organism. We then went through specialized cells as the building block for multicellular organism and discussed the unique features for specialized cells. We then looked at how specialized cells are made and the role a stem cell plays and how a stem cell is converted from what it is into a specialized cell. Now, there is a lot of biology that goes into the differentiation of a stem cell, and we haven't really covered it in this lesson. However, if you do want to extend yourself, you can look up Khan Academy differentiation, as well as there are a number of textbooks in the library that will explain the process of differentiation. But for a year 11 level, understanding that a stem cell turns into a specialized cell when exposed to chemicals and how that happens, that's enough understanding for a year 11 level. Finally, we then looked at where stem cells are found in animals and where they're found in plants. Now, it's important to know that in animals, stem cells are very abundant during the embryonic stages. So once the zygote, the fusion of the egg and sperm, has undergone, there is a rapid division into a blastocyst that contains a large number of these pluripotent stem cells that can be differentiated into a wide range of specialized cells. Whereas in developed animals, so post-embryonic, you don't have these pluripotent stem cells anymore. You have multipotent and unipotent stem cells found within the bone marrow and brain tissue, and these can become a limited number of specialized cells. In terms of, I guess, a bit of extension information, current biological research is looking at creating or artificially creating pluripotent stem cells or using them to treat a range of diseases because they can be turned into specialized cells. So, for example, if you had a disease where your cardiac muscles aren't functioning properly, one of the current big ticket items of biological research is, can we get these pluripotent stem cells and regrow cardiac muscles and embed them over your cardiac muscles that aren't functioning properly? It's a really exciting field, and if I strongly encourage you, if you are interested in it, to look up some papers or textbooks on stem cell research, because it is a very big area for biotechnology, and we will be looking at it again in year 12. Finally, we then looked at where they're found in plants, that yes, there are stem cells in plants and they're found at the root tips for plants. And that sums up our our first lesson for Module 2. If you have any questions, please write them down. I know there's a lot of content covered, and you can come and ask myself or another biology teacher to get a bit more of an understanding. In terms of where to go now with this, I've put up two sets of questions. The first is a PDF that has a very basic breakdown of the analogies of differentiation and specialized cells. And the second one, which are kind of more advanced questions, are exam style questions relating to the content that we have just studied. So please have a go at those. Have them completed. I'm happy to look at them for feedback, or you can come and ask questions when you see me next. Thank you very much, and thank you for listening.